So, ladies and gentlemen, we'll get started with the next set of talks. We have 4 company presentations followed by Jan Lotwal's talk. So we want to make sure we're trying to scale schedule. The first talk will be presented by Dr. Jean-Luc Fraikin, who's the CEO of Spectradyne. And Dr. Fraikin is going to speak virtually. So we'll get started. Welcome, Jean-Luc.
Thank you, Inal, and thanks for organizing such a cool program. I'm looking forward to hearing more of the talks today and tomorrow. Thanks to everyone else who's attending too. I wish I could be there with you today, but not this time, next time maybe. As you know, methods for characterizing EVs are evolving, and new techniques are being developed that measure EV size, concentration, and specific information about the cargo they carry. At Spectradyne, we feel it's important for researchers to understand how the analytical tools that you're using fundamentally work. And that's to empower you as scientists to make more informed decisions on how to interpret the data that you're collecting and ultimately do science faster and easier, right? Which is what we all want. So today, I'd like to share Spectradyne's understanding of how some of these new and evolving techniques work. What that means in terms of strengths and limitations, and where we see the technology landscape moving next. So in my talk today, I'd like to encourage you to think about 3 things. First, that you should take particle size and concentration measurements seriously because they're critical experimental parameters, and controlling them can better help your science. Second, I'd like for you to be asking yourself if your measurement technique is doing a good job of delivering the information you're asking it to. So another way of asking that is, making sure that you're operating within appropriate bounds for the technology. Finally, I'd like to ask you to think about what you feel the field needs next in terms of EV characterization technology, and I'd love to hear your ideas in the Q&A after. So I'll start by making the case for why EV size and concentration are such important parameters to measure. Then we'll briefly review common EV techniques, including Spectradyne's technologies, so we're all on the same page about how they work. Excuse me. Then we'll discuss 3 emerging technologies that deliver some form or another of single particle phenotyping, which is the future. And finally, we'll look ahead from this perspective and summarize where we feel the current needs are. So let's get started. EV size is important. Okay, and that sounds like an obvious statement, but let me explain in a little more detail. So particle size really affects the particle's activity in whatever system it's operating in. So, excuse me, frog in my throat. Particle size determines the diffusion and clearance rates of EVs in biological systems, for example, and how quickly they can get from one place to another in a system to effect a change. That in turn affects the bioavailability of these particles. And so if we're thinking about therapeutics, EVs as therapeutics, this is directly tied to the efficacy of the therapeutic, right? Another area that size impacts directly is the volume of the particle. And this is important because the volume of each particle is the amount of payload it can carry, right, or information or therapeutic active ingredient, right? And that is effectively the per particle dose delivered by each particle, right? So this helps to explain why size is so important. In the case of a therapeutic, it's critical for safety and efficacy of the drug. What about concentration? So and in this case, the overall concentration of particles in the sample really is the dose of the drug or the amount of active ingredient or the amount of information that's being carried, right? And so in the case of EVs as therapeutics, for example, in a wound healing experiment or something, right? The concentration of the particles that are being delivered is the dose, and carefully controlling that, measuring it accurately, enables clearer trial outcomes and ultimately safer, more effective products. So concentration is clearly a critical experimental variable also. This may seem obvious, but let's walk through this from an experimental perspective. Okay, so suppose you have created two different samples, A and B. These are EV formulations, maybe, or maybe they're EVs that have been extracted from a system, and you want to compare their effect on a target system. Maybe this is a therapeutic, or maybe you're understanding the biological activity or role of these different types of EVs. So you prepare the formulations, you apply them to a test system. This might be a live animal model, for example, or tissue culture. And then we want to make a quantitative comparison between the outcomes of these two, of adding these two materials to the test system, right? I don't think there's a scientist in the room or on the phone call that would try and do this experiment without first controlling for the dose, the amount of material that is applied to each system. So this is an experimental example where to highlight the importance of measuring accurate size and concentration, it's really critical for good science. Another application where this comes up is when we're measuring EVs as an assay readout, for example, in biomarker detection experiments. So in this case, the concentration of the EVs really is the test result. You're trying to, maybe there's a biomarker, maybe you're trying to assess the relative amounts of the abundance of different EVs in a system. What you're really trying to measure is quantify each one. So measuring the concentration is clearly a critical variable in this kind of experiment, and measuring them carefully improves the assay development process. So how are people measuring EVs these days? Well, for a long time, cryo-EM has been the gold standard for identifying EVs. It's great because you get to see what you're looking at, right? It's the gold standard for imaging EVs. Of course, it's not practical for routine use. Nanoparticle tracking analysis has been really a foundational technology for the EV field in general, I would say. And so we'll talk about that. This is an optical method, which I'm sure you're familiar with, that measures single particles in the sample. In terms of non-optical techniques, that's where Spectradyne is a fan. There's been tunable resistive pulse sensing for a long time. This is an electrical method, which is a good approach, although I think the landscape shows it's maybe a challenging implementation. And then that's where Spectradyne comes in. So Spectradyne's technology is microfluidic resistive pulse sensing, MRPS. It's a non-optical technique that is single particle measurements and fast, essentially. So let's get on the same page about how it works really quickly. This is a picture of our instrument. It's called the NCS-1. It's a benchtop instrument, and it delivers accurate concentration and size measurement starting from about 50 nanometers diameter up to 10 microns in diameter. As I mentioned, it's an electrical method. I'll talk about how it works next. And it's fast and easy to use. One of the features that our customers really like about it too is that it's microfluidics enabled. That means just a few microliters of the sample are required for each analysis. So how does it work? It's based on the Coulter principle, which has been around for decades and has been the gold standard for measuring particles and cells, large particles and cells for a long time. And it analyzes particles one by one, measuring their size and concentration about as directly as you can. So it works like this. We flow the sample through a bottleneck in the fluid channel, okay, in a microfluidic cartridge. And every time a particle goes through this constriction, we get an electrical signal from which we measure two sort of fundamental properties. One is how big the particle is. So the size of the signal relates to how large the particle is. And then the other thing we measure about each event is how fast it went through the constriction, because that tells us the volume of sample that we've measured. It tells us the flow rate. With these pieces of information, we can measure absolute concentration. So if we measure for longer, you can see many, many signals. They're all highlighted by red dots here. And there are a few different sizes of events, right? Those are different sizes of particles in the sample. And directly from, we can see that directly from the count histogram of events here, right? But we can do better than just counting because we know the volume of the sample that's been measured. And so we can convert these counts to actual absolute concentration. That's what this plot's showing. This is a typical output from our technology. It's got the concentration of particles on the y-axis, and the size of the particles in effective spherical diameter on the x-axis. So you can see this sample, this is the analysis of the data I just showed you, shows 4 populations of particles. This was a mixture of polystyrene beads, 50, 90, 120, and 150 nanometers in diameter. And you can see that our technology is able to resolve them all and be quantitative over any size range here. So this combination of accurate concentration and size together with the ease of use of our platform just is not currently available with any other method. So let's move to the second goal for my talk to get you to think about whether your measurement method is doing a good job in the way you use it. And so the other way to ask this question is, how well do I know the limitations of my technology? And as we all know from the MySEF guidelines, the answer is to use orthogonal techniques, right, or complementary techniques. So let's look into these a little bit more, and starting with NTA, because regardless, many people still use this technology, as shown, by this recent poll. So it's ubiquitous for EV analysis, right? So how does NTA work? I think it's important to get on the same page from here. So we start by illuminating the fluid sample that contains particles with a laser, and we do this in bulk, so many particles are being illuminated at once. The particles undergo Brownian motion, they're not standing still, and so they exhibit these random walks around in space, right? And what we do is NTA monitors these particle tracks with video by detecting the scattered light. So the light comes in, it scatters off into the camera, right? And from the size of these random walks, we can infer the particle size of each individual particle. So this is really cool technology, and it works well for monodispersed samples, but there's a fundamental limitation that limits this for polydispersed samples. So let me explain. The issue is that the scattering intensity from an individual particle depends on the diameter very strongly of the particle. And so what that means is, if we say that a 50 nanometer particle scatters 1 unit of light, then a 200 nanometer particle, which is not that much bigger, scatters 4,000 times much more light. So in practice, what this means is that the big particles are so bright in the video images that they obscure the view of the small particles in the sample. And so the relative abundance of small particles in the sample is undermeasured. Now, this is something that you can test very easily yourself, and so I suggest you try this to understand your technique. So what I'm showing here is a measurement of 3 different samples, 3 separate samples by NTA. This is a 200 nanometer, 150, and 90 nanometer particle. And you can see that NTA measures each one of these samples accurately on its own, right? These were mixed to be about the same concentration. The sizes are coming out just right. And from this, you would reasonably conclude that the sensitivity limit of this technique is somewhere below 94 nanometers. And you'd be right for monodispersed samples. In fact, there are examples of detecting 10 nanometer gold nanoparticles with NTA, right? So that's great. The complexity comes when we start looking at a polydispersed sample, and we can generate one just by mixing these 3 particle sizes together in equal parts in the sample. When we mix them together and measure the sample, this is what the NTA reports. And what you see is that the 94 nanometer particles are completely omitted from the measurement. And the reason is because of what I explained before, is that the large particles in the sample are dominating each video image, and the small particles are too dim to be detected. So effectively, what this experiment shows is that the sensitivity of the technique depends on the composition of the sample, right? And that's a challenge when you don't know what's in your sample to begin with. So as good scientists, let's do the same experiment with an orthogonal technique using MRPS. So again, MRPS measures the monodispersed components accurately, right? All about the same concentration, all the right size. But this time, when we mix them together and measure them by MRPS, we get exactly what we expect, which is the superposition of monomodal distributions of each of the different components, right? So this example is to show the importance of orthogonal techniques and understanding the limitations of your method for measuring samples, right? Measuring EVs. but this is just polystyrene particles, right? So let's look at an example in the real world of extracellular vesicles instead. So this is real NTA data of an EV sample that was measured. And if you look at this data, you might be getting kind of excited. Look, there's a promising peak in the size distribution, right around 130 nanometers diameter. It's easy to track the position of this peak and to say, well, let's, what if we change the formulation a bit? What happens to that position of the peak? But you're a careful scientist, so let's keep going and measure with an orthogonal technique. First, let me replot this same data, okay, except on a log Y scale, so we can resolve differences elsewhere in the spectrum, okay. And now let's measure the same sample using MRPS. So we don't see a peak in the distribution here, and instead, what you see is the concentration of particles increases all the way down to the limit of detection of the MRPS technique, 50 nanometers or so. So what's going on? Well, if you're a really careful scientist, then you go down to the EM core and you measure the same sample by cryo-EM. And what you get there is the exact same particle size distribution as MRPS. They're in excellent agreement. And what we can conclude from this is that NTA's sensitivity to the small particles in the sample rolls off because of these bigger particles in the sample. They're obscuring the detection of these small ones. And this is a critical size range for EVs. Obviously, it's right in the bread and butter size range for where you want to be measuring your particles. So the bottom line here is that orthogonal measurements give really important knowledge about the limit of detection of a technique. NTA works really well for monodispersed samples. That's great. But when we get into real world samples, then it's important to use orthogonal techniques to make sure you're measuring what's really in the sample. Now, obviously, I come from a biased perspective, right? But you don't have to take my word for it. There's plenty of literature that shows exactly this and other really important comparisons between technologies for doing this. And I would highlight some from AMC. Edwin has a lot of great technical papers about this. And this paper from Johns Hopkins from last year that characterized 4 new emerging particle analysis techniques for EV quantification. It's a good segue now then into the emerging technologies. So let's get into that a little bit. Very popular right now is just NTA with fluorescence imaging, right? This is again essentially same method. We're estimating the size from diffusion parameters of the particles, but now we're detecting the fluorescence emission in this technique. Another really cool technology, I think, is single particle interferometric reflectance imaging sensor, or SP-IRIS. This can be coupled with fluorescence. It's another optical technique, but this one allows identification and relative quantification of subpopulations in samples, which I think is definitely the direction that we need to be going in the field, which we'll talk about. And finally, I'd like to talk a little bit about flow cytometry, because this is an emerging method for small particles and EVs, another optical method that I think is the real future of the space. So let's take each one of these in turn. So fluorescence NTA, it's basically the same question, right? Is that it's important to know, like for any technology, what the limit of detection is for the method you're using, right? So let's do a thought experiment. Okay, this is hypothetical data, but suppose you measured this size distribution from your fluorescence NTA. Now you might be thinking, well, this has got to be a real peak now, because we're no longer dependent on the scattering intensity of light in order to detect these small particles. It's just, if they're bright, we'll see them, right? And so what I would ask is just the same question. Ask what the limit of detection is for the technique. So is it true that there are no fluorescent particles here in this size range, or is it just that they're too dim to be detected by the fluorescence detector now in the system? So the point here is to think about that for accuracy of measurement, it's really important to know the limit of detection for size, limit of detection for fluorescence if you're going to be using fluorescence as your detection signal. And then that couples in other questions about how long the samples are stable and stuff in the method, which we won't get into today. Okay, so let's move away from fluorescence NTA and talk about SB Iris, because this is really cool technology. So in this process flow, we start by functionalizing a surface with capture antibodies, right? And then we immobilize particles from the sample onto this surface, and they bind, where the appropriate receptors are. Then this is where the really cutting edge aspect of the technology comes in. The technique uses interference imaging to measure particles that are below the wavelength of light, which is really cool and powerful. This enables sizing of particles, very small particles, and the relative counting and phenotyping by virtue of the specific capture of the antibodies on the surface. That's great. And then you can further couple this with fluorescence to co-locate surface markers on here. And so this is a cool technology for assessing the relative abundance of different particles in the populations, right, by type. Let's look in detail at one of the core processes in this assay, because I think it's an important aspect in terms of interpreting the data. So in this process, the particles are diffusing to the surface in order to be captured, right? And so let's say they have to diffuse a certain distance L, right? The amount of time required for the particles to diffuse to the surface depends on how far they have to go. So it's proportional to L squared in this case. So it takes a while, but that's all right. And then, but it gets complicated because this diffusion time also depends on the size of the particle. And let me point out an example here. So suppose we set L to be a millimeter. So it's like the depth of a droplet of your sample on the chip, or maybe up to a cover slip or something. It takes about 4 hours for a 50 nanometer particle to diffuse that distance in the sample. But this time depends on the diameter of the particle. So a 200 nanometer particle, which is not that much bigger, takes 16 hours to go the same different distance. So as this complicates things, because after you set a fixed incubation time, as a result, large particles interact a lot less frequently with the surface, and they end up being underrepresented. And so the important part for this surface, for any surface immobilization assay, is to understand that it's really, because of this, it's so complicated. It's limited to the relative concentrations of different markers, right? It's hard to get the size information from here. So let's move to flow cytometry now, because I think this is where the field is going. This is a really powerful technology. It's been around for a long time, and it's a foundational technology really for large particle and cell analysis. It is ubiquitous and super well characterized, and importantly offers in situ analysis. So it doesn't require the particles to be immobilized anywhere while they're being analyzed. It's also compatible with standard protocols and labeling. There's very little bleaching concerns, for example, with this technology. And you could argue that a lot of the standard protocols and labeling processes were actually developed for this technology, right? So it's very powerful this way. Let's go through how it works, right? So particles are flowed through a flow cell, and they're illuminated one by one or multiple lasers, right? These systems are super well developed now. And then they would detect the light that comes off the particles with detectors. And there are two types of scattered light that can be detected: forward scatter, that scatters in a forward direction off the particle, and side scatter. And we can also, if we illuminate with an appropriate excitation light, we can look at the fluorescence light that gets emitted from each individual particle as it goes through the flow cell and the beam this way. And these days, flow cytometers are approaching spectrometers where there can be so many fluorescence channels that you really get a very narrow band profile of the fluorescence properties of these particles. It's really impressive technology now. Now, what mechanism are we going to use to count the particles? So to detect them, right? So there are fundamentally two options here. One is to use the scatter pulse, either from the forward or side scatter or both, to detect particle events going through here, right? Or we can look at the fluorescence, at a pulse in the fluorescence channels to detect, say, hey, that was a fluorescent particle that went through. What are the limitations of this method? Well, so counting particles by scatter is limited in a similar way to the other optical scattering techniques that we talked about already. So EVs are small particles and they're low contrast, index contrast with the surrounding water usually, right? And so they're weak scattering particles. As a result, I would say, my understanding is that conventional flow cytometers have a limit of detection in the 1 to 200 nanometer size range using scatter for detection. And I know this is a huge, intense area of R&D for many cytometer companies, so this is changing as we go. What about counting particles by fluorescence? Well, so this has the advantage you can detect smaller particles as long as they're sufficiently bright, right? But comes up against another wall, which is how brightly can you make those small particles? So there are steric limitations for how many fluorophores you can put on a small particle, and that's pretty dramatic when we get into the EV size range. What about measuring size? So with these two, we're talking about the detection method. What about measuring size? Well, so from the fluorescence is not very well defined without using special proxy assays. So for example, if you detect a brightness of a particle, you don't know if it's from a small, very bright particle, like an aggregate of fluorochromes, or a large dim particle, right? So it's hard to get size information from this without some assumptions. And I know there's also intense effort here. What about from scatter? So the scatter light is dependent on the particle size, but requires knowledge of the index of refraction of the particle. And I know there's also a lot of effort here. There's a hot area using flow cytometry for EV characterization, and we have experts in the room about that, so I need to be a little bit careful. But I will make the point that knowing the index of refraction of the sample is not that straightforward. So this is a plot from a paper a couple of years ago from Beckman-Coulter, which they measured a bunch of different particle sizes and on their Cytoflex instrument. And these particles have different classes of particles. There's polystyrene beads in green, viruses in yellow, and silica particles in blue. And they're all of a known size measured with another technique. And what I want to show here is the challenge of using scatter on its own to infer the size of the particle in a complex sample. So suppose we get a scatter intensity signal of 1/5. You don't know in advance whether it's a 100 nanometer polystyrene particle or a 150 nanometer particle that's an HSV virion, or a 200 nanometer silica particle, right? I know that this is where I have to be careful. I know that there are really cool efforts to combine scatter, like forward and side scatter, to try and collapse these curves. It's my understanding they're limited a little bit in which size range those are applicable to, but I'm looking forward to watching that develop. My main point here is that even if you do know the scatter here, determining the optical, the index of refraction of a particle is kind of still a hot topic. It's not totally straightforward. And that's evidenced by the chain of articles that stemmed from this paper in 2019. As recently as December, there's the subject of back and forth between Beckman and researchers in Amsterdam about the interpretation of exactly this kind of data and the modeling of the particles in different ways to assess their refractive index. So it's a complicated topic and is, it's complex, right? So to summarize, flow cytometry, for optically homogeneous samples, obviously that's great, where we know the index of refraction. To get size in heterogeneous samples, I think everyone would agree it's a challenge to get EV size, and work still needs to be done to make that robust. And in the meantime, we need orthogonal techniques in order to measure the actual size and concentration of the particles in the sample. So let's look ahead now. What's next, right? I hope I've convinced you that there are many techniques out there for measuring concentration and size directly. but I think we can agree that more in-depth EV analysis demands not just size and concentration, but also phenotyping, right? And real concentration, not just counts. And that there's still a critical need for single particle phenotyping techniques with accurate size and concentration and well-characterized limits of detection. And so what I mean by that is limits of detection in size, concentration, and fluorescence intensity in terms of a relatable standard unit like MESF, for example. Of course, these should be, fast and easily integrated with standard reagents and methods and stuff. Okay, so I'm interested in hearing from you. We have a couple minutes left. Just in conclusion, I would like to say I hope I've convinced you that accurate size and concentration are critical for good science. I've gotten you to think about the limitations of the technologies you're using and making sure that you're operating within the normal or the appropriate bounds for those technologies, and that there's still an unmet need out there for accurately quantifying subpopulations in complex samples. If you'd like to learn more about Spectradyne's products, please visit our website. We have a large and growing library of publications and application notes. And we encourage you to get in touch with us and send us a sample. So thank you very much for your attention today, and I hope you enjoy Rotterdam.
Thank you very much, Jean-Luc.